Hello dear students, in last class we have learned about pollination, the types of the pollination and pollinating agents. Today let us learn about pollen pistil interaction, fertilization and other post-fertilization events. What is the pollen pistil interaction? Pollen pistil interaction is a group of events that occur from the time of pollen deposition over the stigma to the time of pollen tube entry into ovule. It is an essential step in fertilization of angiosperms. The pistil has the ability to recognize and accept the right or compatible pollen of the same species, and pistil also can reject the pollen grains that are incompatible either of the same species or of other species. After recognition of the correct pollen, pistil promote post-pollination events that leads to fertilization. The compatible pollens are able to absorb water and nutrients from the surface of the stigma and germinate to produce pollen tubes. Their growth and path through the style is also determined by specific chemicals like calcium, bromine, etc. Pollen pistil interaction is mediated by chemical components of the pollen interacting with those of pistil. It involves the following events. Firstly, following compatible pollination, pollen grain germinates on the stigma to produce a pollen tube through a zygote. Pollen tube grows and reaches the ovary, enters the ovule through micropyle and then enters one of the synergids. These are the steps of pollen pistil interactions, right? Now let us know about artificial hybridization. What is artificial hybridization? Artificial hybridization is a type of crossing that is performed by human being. Two important techniques which are used in hybridization are emasculation and bagging. Emasculation is the removal of anthers from the flower bud before the anther descends using a pair of forceps. What is bagging? It is the covering of flower by butter paper or polythene before the flower opens. The emasculated florent buds of the female parents and the dead florent buds of the male partners are bagged in order to protect from contamination of unwanted pollen grains. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are back before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen grains, and then the flower re-bag. Now let us know about the fertilization occur in angiosperms. Fertilization is the fusion of a male and a female gamete to form a zygote, right? In seed-bearing plants, the male gametes are brought to the egg-containing female gametophyte or embryo sac by a pollen tube. Such fertilization with the help of the pollen tube is called Siphonogamy, right? You know, in flowering plants, the fertilization is known as double fertilization because two types of the fusions occur in flowering plants. Out of the two male gametes carried by pollen tube, one fuses with the egg cell to perform generative fertilization, which is also called syngamy or true fertilization. It forms a diploid shell, which is known as a zygote, right? So the second male gamete fuses with the polar nuclei to form a triploid primary endosperm nucleus, PEN. This second fertilization is called vegetative fertilization, since it produces a vegetative or nutritive tissue to nourish the embryo. Second fertilization is also known as triple fusion because three nuclei get fused, two polar nuclei and one male gamete. Double fertilization was discovered by Nawasin in 1898 in Fritillaria and Lilium. So now let us know about significance of double fertilization. Double fertilization ensures the formation of endosperm only when the egg has been fertilized and the embryo development is to start. Due to triploid in nature, endosperm shows high physiological activity, grows faster, and accumulates nutrients. So now let us know about development of endosperm. Endosperm is a triploid and it develops from primary endosperm nucleus. The primary endosperm nucleus undergoes repeated mitotic division without cytokinesis. At this stage, the endosperm is called pre-nuclear endosperm. Cell 1 formation occurs and endosperm becomes a cellular type. The number of free nuclei formed before cellularization varies greatly. Example, in coconut. In coconut fruit, the coconut water present in the sheath is a free nuclear endosperm and the surrounding white kernel is cellular endosperm. Actually, there are three types of the endosperms based on the type of the development. First type of endosperm is called nuclear endosperm, which we have learned now. Next is the cellular endosperm. In cellular endosperm, each and every nuclear division is followed by cytokinesis. So endosperm is cellular from the very beginning. Example of cellular endosperm is found in Datura and Petunia. Third type of endosperm is called halobial endosperm. It is an intermediate between cellular and the nuclear endosperm. The first nuclear division is followed by cytokinesis to form unequal cells, micropylar cell and Salazian cell. Further divisions in each cell are free nuclear divisions. The type of endosperm is found in Espodellus. Now embryo formation. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Mostly, zygotes divide only after certain amount of endosperm is formed. The nutrition for the development of embryo is provided by the endosperms. The zygote gives rise to pro-embryo and subsequently to the globular, heart-shaped and mature embryo. In a typical dicot, the zygote elongates and then divides into two unequal cells. The larger basal one called suspensor cell and the smaller one called embryonal cell. The suspensor cell which lies towards the micropyle and divides transversely a few times to produce a filamentous suspensor of 6 to 10 cells. The suspensor helps in pushing the embryo in the endosperm. The embryo divides to produce Octane embryo, which are arranged in two tiers. 4 cells near the suspensor are called hypobasal, and other 4 cells are called epibasal. Epibasal cells eventually form the cotyledons and epicotyles, and hypobasal cells produce hypocotyles. Tips of the hypocotyle is called radicle. So let us see how embryo develops in monocot plants. Embryo or monocot possesses only one cotyledon at one end, and the cotyledon is called scutellum. At lower end, the embryonal axis has a radicle and the root cap enclosed in an undifferentiated sheath called coleorrhiza. The portion of the embryonal axis above the leaven attachment of scutellum is the epicotyle. Epicotyle has a short apex and a few leaf primordial enclosed in a hollow foliar structure called coleoptile. You know, some examples of the monocot plants are grass, banana, and bamboo. For both dicot and the monocot sheaths, upper embryonal axis is called epibasal and lower embryonal axis is called hypobasal. Now let us know about sheath. The ripened ovule after fertilization is called sheath. A typical sheath consists of a sheath coat, cotyledon, and an embryonal axis. The integuments of ovules get transformed into sheath coat, outer testa, and inner tegment. The micropyle of the ovule is sensed in the micropyle of sheath through which oxygen and water enter the sheath at the time of germination. The wall of the ovary becomes a pericurve. Pericarp is divided into three parts. Outer part of pericarp is called epicarp, then mesocarp, then endocarp, right? So let us know about the types of the sheaths, which is based on presence and absence of the endosperms. In some sheaths, the endosperm persists in the sheath as the food storage tissue. Such sheaths which are having endosperms are called endominous or endospermic sheaths. Some examples of endospermic sheaths are castor, maize, wheat, barley, sunflower. Okay, so next type of sheath is called exalbuminous or non-endospermic sheaths. In such type of the sheaths, the endosperm is completely eaten up by growing embryos, and the food for later development of embryo is then stored in cotyledon. Such sheaths are exalbuminous or non-endospermic sheaths. Examples of non-endospermic sheaths are pea, gram, bean, etc. In the dry sheath, the embryo persists in the state of inactive, called dormancy. In some sheaths, such as the black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucellus is called perisperm. Okay, let us know about fruits, types of the fruits. Actually, fruits are developed from the ovary, right? So the fruits developed from ovary are called true fruits. If the fruits are not developed from the ovary, but they are developed from other parts like thalamus, this type of fruits will be known as false fruits. Some examples of false fruits are found in apple, strawberry, and cashew. The seeds of apple, strawberry, and cashew are false fruits because the fruits are developed from thalamus, not from the ovary. So next, let us know about parthenocarpic fruits. Some fruits develop without undergoing fertilization. These are called parthenocarpic fruits. Examples are banana, grapes. Parthenocarpic fruit can be induced through the application of growth hormones like auxins and gibberellins, and such fruits are seedless. Now let us know importance of seed. Firstly, seeds have adaptive strategies to get dispersed to new habitats and colonize as the same. Seed has a sufficient food reserve that nourishes the germinating embryo and growing seedling till it becomes photosynthetically independent. Being products of sexual reproduction, seed have a number of variations due to genetic recombinations and crossing over. So next is seed viability. What is the seed viability? The functional period of seed is called seed viability. There are several records of very old yet viable seeds. The seeds of lupine. Scientific name of lupine is Lupinus arcticus, excavated from Arctic tundra, are the oldest. The seed germinated and the flower after an estimated record of 10,000 years of dormancy. And the seeds of Phoenix dactylifera, discovered during the archaeological excavation of King Herod's palace near the Dead Sea, have a record of 2,000 years old viable. Right? Let us know about apomixis now. Apomixis is a modified form of reproduction in which seeds are formed without fertilization, without fusion of gametes. It includes the process whereby a diploid cell of nucellus develops into an embryo, giving a diploid seed with a genetic constitution identical to the parent. The organism that reproduces by apomixis is called apomic. Next is polyembryony. What is polyembryony? Occurrence of many embryos in a seed is called polyembryony. Right? Occurrence of polyembryony due to fertilization of more than one egg is called simple polyembryony. Formation of extra embryo through sporophytic budding is called adventive embryony. Polyembryony is quite common in onion, groundnut, mango, lemon, orange. Okay, dear students, let us learn this much for today.